Our next speaker of the day is Jocelyn Armistead, who is from Brock University, and she will be given a presentation about a comparison of collection methods for evaluating Canadian bumblebee diversity. Go ahead, Jocelyn.
Thank you, Nora. My screen should be sharing now, I believe.
Yes.
Thank you. Okay, so yes, I'm Jocelyn Armistead, and I'm a master's student at Brock University. And I'm going to be talking to you today about a comparison of methods for evaluating bumblebee diversity. So researchers are often interested in conducting assessments of bumblebee abundance and diversity in order to understand how populations are changing. For example, in Canada, COSEWIC conducts assessments for species thought to be in decline, and they've done assessments for a number of bumblebees. To evaluate abundance and diversity, bumblebees need to be counted, but currently there's many different collection methods being used. And so we're interested in finding out if there is a best method. The qualities of a good collection method are that it should produce accurate reports of bumblebee diversity and species composition, detect as many species as possible, including rare ones, work well across regions and environments, be simple and cost-effective, and non-damaging to populations. So the objectives are to evaluate 3 collection methods to assess whether they produce different bumblebee diversity and species composition results, to evaluate the collection methods for the level of sampling effort required, and then based on the outcomes from the first 2 objectives, to provide a recommendation for which methods should be used in assessments of bumblebee diversity. So in 2021, surveys were conducted in 3 regions of Canada. I did the surveys in Ontario in the Carolinian zone. Corey Sheffield's team did the surveys in Saskatchewan in the Prairies ecozone, and Carolyn Parsons' team did the surveys in Newfoundland and Labrador in the Boreal Shield ecozone. While there were some differences, the overall structure of the surveys was similar across all regions. For instance, all 3 regions used the same collection methods. So blue vein traps, which are shown in the picture on the left, they were either placed on stands or in the ground, and they are typically deployed for a week at a time. Targeted netting used an insect net to collect bumblebees for 30 minutes per collection event. And photographs were done using cell phone cameras for 30 minutes per collection event, with multiple collection events done throughout the survey week. And so we'd like to know what the different collection methods tell us about diversity and species composition. So how many species were detected? Were any of the at-risk species detected? What the species composition is, and for that we'd be looking at the relative abundance, which is the proportion of the sample that a species makes up, and as well PLO's evenness, which is the level of similarity across those relative abundances. And additionally, we're interested in knowing about the collection efficiency, so the number of species per individual collected. The method that collected the most species varied by region. In the Carolinian and Prairies ecozones, blue vein traps collected the most species. Particularly in the Prairies ecozones, blue vein traps collected many more species than the netting and photo methods. However, in the Boreal Shield ecozone, netting collected the most species. And then this table here has a list of bumblebee species that have been assessed by COSEWIC and potentially could be found in the regions that we surveyed. And then the table shows a combination of results from all regions. So if a method detected a particular species, even in one region, it has a check mark here. And overall, what we can see is that blue vein traps detected the most species at risk. Now looking at how the species composition varied, these figures show the bumblebee species along the x-axis, and then the relative abundances along the y-axis. And relative abundance was calculated per collection method. And then those collection methods are shown in the 3 different colors. And comparing across all 3 regions, a pattern emerged where the netting and the photographic methods produced a much more similar species composition to each other than to the blue vein traps. And we can see this in all 3 regions, or blue vein trap distribution is the most distinct. To provide some statistics for this, I ran chi-squared tests, and indeed, for all 3 regions, the chi-squared indicated that yes, the species composition varied significantly by collection method used. Now, one of the reasons for this significant difference is the blue vein traps consistently collected a much lower proportion of the most common species in each region. And overall, this resulted in a more even distribution, which can be seen particularly here in the Carolinian zone. And calculating PLO's evenness, we do see that blue vein traps consistently had the highest evenness. So now looking at the second objective, which collection method requires the lowest sampling effort while still detecting as many species as possible? And we're interested in the method that has the lowest sampling effort because it requires less time and it produces less bee mortality. So these figures here are species accumulation curves that show how species richness increased over the collection season. Along the x-axis is the collection date, and along the y-axis is the cumulative species richness. Each point is a collection date, and so we can see on which dates new species were detected for each method. And looking across all 3 regions, what stood out is that blue vein traps were the only method that continued to collect new species throughout the entire collection season or most of the collection season. Whereas the other methods all reached a plateau at some point and stopped detecting new species. You can particularly see that in the Carolinian zone. Now, these figures are rarefaction curves, and rarefaction uses subsampling to create estimates of species richness based on the number of individuals, and it's useful for comparing collections that have different sampling efforts. So along the x-axis is the number of individuals in the subsample, and along the y-axis is the cumulative species richness. And the main pattern I'd like to point out is that blue vein traps detect more species with fewer individuals. So we can see for all of the figures, the red curve is above the green and blue curves at a low number of individuals. And in the Carolinian and in the Boreal Shield ecozones, blue vein traps collected far fewer individuals than the other methods. However, in the Prairies ecozone, blue vein traps actually collected many more individuals than the netting photo methods. However, they are also detecting the most species as well. And the dashed lines indicate the estimate of species richness based on if sampling was continued past what we did in this study. And from that, we can see that for the Carolinian and the prairies ecozones, blue vein traps are expected to detect the greatest species richness. And for the boreal shield, it is the netting method that's expected to find the most species with continued sampling. So to summarize, which method was best for determining diversity in species composition? Well, the method that detected the most species varied by region. In the Carolinian and prairies ecozones, the blue vein traps detected the most species, but in the boreal shield ecozone, the netting method detected the most species. Overall, though, blue vein traps collected more of the species at risk, which suggests that blue vein traps might be more useful for studying these species. Species composition varied by method used, and we did, however, see a pattern, though, that the netting and the photo methods produced more similar species compositions to each other than to the blue vein trap method. So which method required the lowest sampling effort while still detecting the most species? Well, blue vein traps were the only method to continue finding new species throughout most of the collection season. As well, they also found the greatest species richness with the fewest number of individuals based on the rarefaction curves. So my overall recommendation is to use blue vein traps, but since they're producing a different species composition from the netting and photo methods, they need to be used along with one of those other methods. In order to choose between the two, it will probably depend on your individual study. Netting worked particularly well in the boreal shield ecozone. So if you're in that region, that might be the method you'd prefer to use. Photographs have the added benefit of being non-lethal, which would be good for working with rare at-risk species. However, it can be a bit harder to identify certain species by photographs alone. And I would just like to say thank you to everyone who has been involved with this project, everyone at the Brock Bee Lab, all of the other surveyors, the private and public property owners, and anyone who provided advice. Thank you so much.
Thank you so much, Jocelyn. We have 2 minutes for questions. Does anybody have a question for Jocelyn? Please put it in the chat box. Oh, there is one. There's actually several ones. One of them is, was there any consideration for baited and unbaited blue vein traps or blue vein traps with unpainted buckets, as shown in your presentation, versus those spray painted neon yellow? Do you think this would have an effect on blue vein trap sampling efficiency?
So, okay, there's a couple parts to that. With the baited versus unbaited, from what I've read, unbaited traps seem to work as well. And you don't have to add a bait to the blue vein trap. It should be the color of the veins that attract the bumblebees. So we had a propylene glycol solution placed in the collection jar, but that was mainly to act as a preservative, not as a bait. And then in terms of the different color collection jars, we used the clear ones for all of the regions that were sampled. I do know that there are ones that have the yellow paint. I haven't worked with them, and I'm not familiar if that would affect the attractiveness to bumblebees or not.
Thank you, Jocelyn. That was Anthony Ayers who did that question, who made that question. There's another question by Jans. Is there a hypothesis why there is a higher change of rare bees in the traps? Since the species seem to be in the area Oh, sorry, that was another question. Okay. So the question was, is there a hypothesis why there's a higher change of rare bees in the traps?
So the rare species that were collected, they were collected in very low abundances. So they quite likely are out in the region, but perhaps we just didn't survey maybe at the timing that they were there, or just by chance we didn't find them. There's a potential that perhaps the traps are more attractive to those species. I don't 100% know the reason why they were found in the blue vein traps and not by the other methods. But I will say that they weren't found in high abundances.
Thank you for that. Sarah McKell has another question. How many netting events were completed throughout the week? In the presentation, it said several, but curious what the average was per week.
So it varied by region. And for instance, in Ontario here, I did 3 netting sessions and 3 photo sessions in a week.
Thank you for that. And I believe that it's all the questions that we have. Oh wait, there's one more, and we have some time for one more question. How do you account for the additional information on flora associations from netting and photos?
How do I account for, you can have the benefit of getting floral associations with the netting and the photo method, but you can't with the blue vein traps. Yes, that's certainly a strength for those methods that the blue vein traps don't have.
Thank you so much, Jocelyn. We'll move to our third